Hello, my name is Egon Cholakian. At this very moment, as you are looking at the screen and listening to this video, the only force that keeps life within your body is an ordered electrical current. Every breath you take, every blink of an eyelid, and even every thought is the result of an electrical impulse. Billions of such impulses occur inside you every second. This is the strict, highly organized electrical architecture of life. Yet today, this electrical architecture is under a concealed but total assault. You have been hacked. If you have recently experienced an unexplained loss of energy, mental fog, memory lapses, or decline in concentration, understand this: it is not just fatigue. These are the earliest symptoms that your electrical code has been compromised. And today, you will learn how this happened and how it continues to happen. Into the finely tuned electrical system of your body have entered, without exaggeration, killers, true agents of chaos, nanoplastics, artificial polymer particles that are hundreds of times smaller than a human cell. Yet when they enter a living biological organism, they inflict damage of staggering magnitude. Every day you inhale, drink, and consume enormous quantities of these nano killers. As they accumulate within your body, they do not merely pollute it or damage individual cells. They penetrate deeper. They disrupt the very electrical code that governs your biology, dismantling the electrical architecture of your organism, the foundation upon which all life itself depends. You will also learn how the biological electricity that sustains your life actually functions, why the flow of electrons in a household power outlet and in the flow of electrons within your DNA are fundamentally different phenomena, and what so-called unique electrical capabilities are concealed within your genetic helix. I will warn you in advance, this is not a conversation for consumers of short, viral mental junk food. If your cognitive threshold is limited to a 6-second clip and your brain demands simplistic, pre-digest conclusions, turn this off. This material is not for you. It is for those who have retained the capacity for deep analysis, for those who still possess the intellectual resources to think information through. In this video, I am speaking to those who are willing to penetrate the essence of the matter rather than skim its surface in order to grasp the real physics of their own survival. If you are capable of such an intellectual effort and ready to delve into the core of the issue, welcome to a reality in which your life hangs by a thin electrical thread, and that thread is now being deliberately severed. Let me begin with a simple analogy. You are watching this video on a smartphone, a laptop, or even a television. You see me on the screen for one reason only. Inside your device at this very second, billions of electrons are obeying a rigid organization. They become excited, relax, and jump from one energy level to another. The key word here is organization. The system functions not because electrons merely move, but because they move according to a strictly defined protocol, from the processor to the circuit board, at a specific frequency along a specific vector. Now imagine that into this orderly chain, into this organized protocol, an extraneous signal intrudes, a kind of parasite, an electrostatically charged object that disrupts the rhythm of the electron flow. As a result, an electron, instead of following the plan, deviates from it. The signal becomes distorted. The processor begins to short circuit. The mechanism breaks down and ceases to function. Your body is, in essence, the same kind of mechanism. All processes within you are likewise regulated exclusively by an ordered flow of electrons. Cell division, enzymatic catalysts, protein synthesis, every contraction, the myocardium, and every neuronal impulse. At the foundation of all this lies a planned, organized movement of charges, electrons, protons, ions. This movement follows a rigid protocol. This is your electrical code, biological electricity that must flow strictly according to plan. As long as this code executes without errors, cells know the algorithm of action, how to divide, how to regenerate tissue, how to eliminate viruses. This is what we call immunity, youth, health, life. But ask yourself this one question. What happens when interference intrudes into this ideal code? Note that we are not referring to a chemical toxin that can be easily eliminated. We are instead talking about an electrostatic noise agent that disrupts the very organization of electrical processes. Nanoplastics penetrating the very architecture of cells, proteins, DNA, and neurons do not merely clog them like dust. They do something far more frightening. They begin to literally rewrite the electrical code, replacing the original program with a false one. To comprehend the mechanics of this destruction, we must descend to the level where biology becomes physics, into the realm of quantum states. However, before I explain in detail the unique electrical processes of your body and demonstrate precisely how plastic breaches them, I will allow myself one important digression. There is a nuance that, as I observe, almost entirely escapes the attention of most people whenever the harm of nanoplastics is discussed. I insist that you first grasp one fundamental fact. Your body does not know what to do with nanoplastics. Listen to this again. Your body does not know what to do with nanoplastics. It does not know. The organism simply lacks any evolutionary protocol for interacting with this agent. Consider this carefully. Human biology was refined over billions of years. During that time, we encountered dust, sand, combustion byproducts, and heavy metals released during volcanic eruptions. Yet all of these were natural compounds. The organism learned to recognize, isolate, and eliminate them. The immune system spent millennia encoding instructions for defense. But in the 20th century, humanity conceived a brilliant idea to synthesize something that had never existed in the biosphere. We created artificial molecular polymers. This means that over 4.5 billion years of Earth's history, this material simply did not exist. And now this absolutely foreign material, plastic, degrades into nanoparticles and enters your body. It penetrates the bloodstream, reaches internal organs, crosses the blood-brain barrier, and accumulates in the brain. And your body quite literally does not know what to do with it. Not a single enzyme, not a single receptor is adapted to break down the synthetic substance. How does nanoplastic manage to overcome the body's most powerful protective barriers? Once inside the body, it immediately forms what is known as a protein corona. This is a dense shell composed of proteins and lipids from your own blood, which completely envelopes the particle. Under the cover of this shell, nanoplastic carries out a covert infiltration like a Trojan horse. It passes through membranes, overcomes the blood-brain barrier, and finds itself deep within cellular structures. And only there, within the cytoplasmic environment, does its disguise actually collapse. The corona degrades, exposing the true electrostatically active essence of nanoplastic. On the surface of a plastic nanoparticle protrude fragments of numerous synthetic polymer chains, which are also electrostatically charged, and with which the body has no idea how to interact. It is precisely this looseness, the complexity of its surface, and its distinctive electrostatic charge that make it so insidious for a living cell, which attempts to interact with it as a single object, but instead encounters a chaotic array of chemical groups and physical properties. For your immune system, nanoplastic is, if you will, an alien, one that masquerades as familiar until it begins its sabotage. And that sabotage consists in the fact that nanoplastics effectively rewrite the internal rules governing your biological system, forcing the organism to function in contradiction to its original design and compelling all key elements of the body, cells, proteins, and acids to behave differently, not as nature intended. I will now illustrate this process using concrete examples. The bioelectricity that sustains life in your body can be conditionally divided into 3 levels. Nanoplastics breach the electrical code in each of them. The first level is ion transport. This is the outer layer of the electrical code. Membrane potentials, sodium and potassium channels. Nature has prescribed a strict algorithm here. The doors of these channels open and close only in response to a specific signal. How exactly nanoplastics disrupt the electrical impulse of your life at this level was vividly demonstrated in the film Nanoplastics: Threat to Life. The mechanism is as follows. An electrostatically charged plastic nanoparticle, as it approaches a neuron, physically deforms the very architecture of ion channels with its electric field. In other words, it literally damages the doors through which charges move. The gates by design are meant to protect the cell, but they stop responding to their natural signal and begin to behave chaotically. They either become jammed shut or fly wide open. As a result, the orderly flow of ions turns into a chaotic draft. The signal becomes distorted, and the code of data transmission in the brain begins to collapse. It is precisely from such disruptions that various degenerative processes originate, including neurodegeneration, Alzheimer's disease, and Parkinson's disease. Let us move further, one level deeper. The second level is electron transport, and the central stage in this process is the Mitochondrial. At this level, nanoplastics attack the key electrical processes occurring within it. Let me remind you that the mitochondrion is a component inside your cell. Its primary function is energy production. What constitutes energy for a cell? It is a specific type of molecule that is produced primarily within mitochondria and then consumed by all other parts of the cell to sustain their function. This molecule is called adenosine triphosphate, or ATP for short. ATP is the specific food, the universal fuel that all components of your organism use to maintain life. Without ATP, a cell comes to a complete standstill. The mitochondrion produces this fuel through biological electricity. The process is complex, but its essence comes down to an extremely precise transfer of electrons. These electrons are obtained by the mitochondrion from the food you consume, specifically from glucose and fats, and are then directed along a strictly defined route, known as the respiratory electron transport chain. At the end of this pathway, according to the natural design, the electron must safely combine with oxygen, O2, to form ordinary water, H2O, which is required for the continued healthy process of ATP production. But what happens when nanoplastics interfere? It has been experimentally established that plastic nanoparticles, by virtue of their electrostatic charge, when entering the mitochondrion, literally knock electrons off their program path within this chain. and an electron that has been forced off course reacts with oxygen incorrectly and prematurely. As a result, instead of forming safe water, H2O, which is required for the cycle that produces the beneficial ATP molecule, an aggressive form of oxygen, O2-, the superoxide radical, is generated. In other words, not only does the process of ATP production come to a halt, but as a byproduct, an extremely dangerous form of oxygen, O2-, also emerges. This is a molecule with an unpaired electron, one that aggressively seeks something to bind to, and doing so begins to tear electrons away from surrounding structures. As a result, it damages them, quite literally destroying everything around it: DNA, proteins, and cellular membranes. In science, this destroyer, O2-, is known as a free radical, and the destructive process itself is referred to as oxidative stress. Oxidative stress is likely one of the most common terms you have encountered if you have come across descriptions of the harm nanoplastics inflict on living organisms. And now you understand what this term actually means. Nanoplastics have interfered with the process of electron transport, disrupted the organization of electrical current in the body. Beneficial oxygen has been transformed into a toxic poison. As a result, the cell dies from a double strike inflicted by nanoplastics. First, it starves because the mitochondrion stops producing its fuel, ATP. This is what science refers to as mitochondrial dysfunction. Second, the cell becomes inflamed due to internal poisoning, oxidative stress caused by O2- free radicals, and the subsequent cascade of dangerous oxidative reactions. And this brings us to the third level of bioelectricity. If the first two levels are well known and thoroughly studied, the third began to be recognized and widely discussed in science only in the early 2000s. This is the quantum level of bioelectricity, one that for a long time remained unspoken of, yet whose existence has now been confirmed by recent experiments. So the third level is spin-dependent electron transport. This is a level of biological electricity transfer in which not only the charge of the electron matters, but also its spin. Here, for a few minutes, we will need to step slightly aside from the topic of nanoplastics. This is necessary so that I can explain to you one unique fundamental aspect of the nature of electricity within you. I believe those who watched my previous address noticed that I mentioned this topic there, namely the special role of electron spin in biological electricity. I received many letters with questions about this aspect. Well, I hope that after this explanation you will have even more questions, and that would be the correct outcome. Because the presence of questions is a sign of an active thirst for inquiry, and where such a thirst exists, answers are never far beyond the horizon. So, regarding spin, many people are accustomed to thinking that electricity is simply the movement of charge, plus and minus. This is the level of school curricula sufficient to make a toaster work, but not to understand life. An electron possesses another property, far more subtle: spin. What is spin? Or more precisely, how does science currently define what spin is? In simplified terms, spin can be understood as a quantum orientation of an electron. At any given moment in time, it has either spin up or spin down. There is no third option. It is important to understand that a real electron is not physically spinning anywhere. However, it possesses these two degrees of freedom, spin up and spin down. And these spin states are directly related to magnetic fields, but that is a separate topic. In an electric current flowing through ordinary conductors, for example, in the wires of your charger, in batteries, in your computer or smartphone, electron spins are distributed 50-50. Half the electrons have spin up, half have spin down. This is because for the transfer of charge, for the energy that lights the lamp in your room, the spin orientations of the electron simply does not matter. However, scientists, especially those studying magnetism, have actively experimented with spin for decades. They asked a fundamental question. Is it possible to obtain a current in which electron spins are not distributed 50-50, but in which electrons with one spin orientation dominate? For example, a current in which predominantly spin-up electrons flow. And what properties would such a current have? By the end of the 20th century, scientists were actively investigating this phenomenon, conducting experiments aimed at obtaining what is known as spin-polarized current, a flow of electrons in which electrons with only one spin orientation are predominantly present. However, to achieve this essentially quantum effect, scientists required extremely specific conditions: ultra-low temperatures, external magnetic fields, ferromagnetic materials, and a carefully isolated environment. It was believed that the quantum state of spin was so fragile that maintaining its ordered state was possible only under rigidly controlled laboratory conditions. As a result, spin-polarized current was long regarded as a technological curiosity. The scientific community was therefore astonished when, in the early 2000s, it became eminently clear that what researchers had been trying to create in laboratories for decades had already existed in nature for billions of years. It turned out that on Earth there is a natural molecule system capable of generating a spin-polarized current directly in a liquid environment at room temperature without the need for magnets and extremely low temperatures. Moreover, this is not a rare exception. There are trillions of such systems inside each of us. They turned out to be the fundamental molecules of life itself: DNA, proteins, and amino acids. These molecules are not quite ordinary. They are chiral. It has been experimentally established that chiral molecules function as spin filters. When electrons with different spin orientations pass through them, such molecules selectively transmit some electrons while scattering others. For example, a molecule may transmit electrons with a conditional upspin and scatter electrons with a downspin. As a result, a spin-polarized current forms at the output, and it turns out that it arises naturally within the chiral molecules of living systems. This phenomenon marked a breakthrough, and in 2011 was named the Chirality induced spin selectivity effect, or the CIS effect, and it is a property unique specifically to chiral molecule. What is a chiral molecule? A chiral molecule is a molecule that has a right-handed and a left-handed form. The word chiral itself comes from the Greek word chir, meaning hand. Humans have a right hand and a left hand. They look similar and are mirror images of each other, yet they are different. The same is true of a chiral molecule. Its right-handed and left-handed variants are mirror images of each other, which means their atomic composition is identical. The chemical formula is the same, but their three-dimensional spatial arrangement differs. As a result, the right-handed version of such a molecule has properties that differ from those of its left-handed counterpart. For example, consider a car. The left door is a mirror image of the right door, but it cannot be installed into the opening designed for the right one. The components appear to be same, yet their functionality depends on geometry. In chemistry, the principle is exactly the same. Different geometry produces a completely different effect. The foundation of life on Earth is homochiral. This means that living systems on Earth are built from molecules of only one mirror type. Proteins are constructed from amino acids of a single handiness. The sugars in DNA are likewise of only one type, and the DNA molecule itself is typically twisted exclusively in a right-handed direction. Mirror variants of such molecules are in principle possible, but in ordinary biology, they are either almost never encountered or appear only in rare and exceptional cases. In this sense, one can say that we are a right-handed civilization at the molecular level. And as it turns out, this is not merely a geometric feature. The cis effect demonstrates that precisely such chiral molecules are capable of selecting electrons based on their quantum state, their spin. Why does this happen? Why are chiral molecules inside our bodies able to generate a spin-polarized current? That is, a current in which electrons with only one spin orientation dominate. Currently, this remains a mystery to science. Numerous theories attempt to explain this phenomenon, including the spin blockade theory, the orbital filter theory, interface enhancement, and the role of dissipation and non-equilibrium processes, among others. However, none of them can fully account for the experimental data. Why? Because experimentally, the chirality induced spin selectivity effect manifests far more strongly than simple quantum mechanical models with spin-orbit interaction predict. This raises serious questions since standard theories yield only small values of spin polarization for single chiral molecules, whereas experiments observe polarization on the order of equal equivalence up to 80% and even higher. This means that in experiments measuring current transmission through a chiral molecule, roughly 80% of the electrons emerge with the same spin orientation. Such values cannot be fully explained within the framework of the generally accepted quantum mechanical models. The gap between theory and practice remains the central open question in the physics of cis effect. After all, it appears that nature is using mechanisms that do not fit within our existing physical formulas and laws. Science can see that it works, yet it does not fully understand why, and why it works with such remarkable efficiency. And why does nature need this mechanism at all? In this area, several hypotheses and experimental observations are available from which researchers draw inferences about why nature employs such a unique mechanism of spin filtration. It has been suggested that a spin-polarized electron current passing through a chiral molecule functions as something like a precision guide, ensuring the safety of chemical reactions and facilitating recovery and repair. Scientific publications note that such an ordered spin current increases the precision of chemical reactions, allowing electrons to move in a more coordinated manner, and just as importantly, prevents random reactions with oxygen. The very reactions I mentioned earlier, in which oxygen transforms into an aggressive form, O2-, and begins to destroy everything around it, oxidative stress. It has been established that an electron flow with an ordered spin orientation helps reduce oxidative stress in DNA. Additionally, such an electron flow plays a crucial role in the self-assembly of proteins, facilitating their correct folding and minimizing errors. It performs a similar function in many other bioelectrical processes upon which the normal, healthy operation of cells depends. One more recent study, which was also mentioned incidentally in the Alatra report on nanoplastics, indicates that spin-polarized current also plays an important role in the proper transport of protons, that is, hydrogen atoms within cells. And all of this represents only the very first signs of an answer to the question of why the organism needs this unique mechanism, the ability to generate a spin-polarized current. This compels us to raise a broader question about the very nature of biological electricity and about what we actually know about it. For decades, physics operated under the assumption that electron spin was irrelevant to biology. Only about 15 years ago did it become clear that this assumption was wrong, and that spin plays not just an important role, but potentially a decisive one. This leads to an unavoidable question. What else do we not yet know about biological electricity, and how transformative might these discoveries ahead prove to be? I would like to invite you to reflect on the following. The organism contains both chiral and achiral molecules. However, the key structural and functional elements of life are inevitably chiral. These include amino acids from which proteins are built, their spatial organization, sugars and carbohydrates, as well as nucleic acids, DNA and RNA, together with the hierarchical structure. All of these chiral structures are the foundation of life. And at the same time, it is precisely the same chiral structures that lie at the center of the remarkable process of generating and transmitting spin polarized current. This means that this type of current and the spin ordering of electrons itself has fundamental significance for life as such, a significance that science has yet to comprehend fully. Drawing on the body of knowledge and research accumulated over more than 20 years by the illustrious scientific community, I can confidently state several key points. First, the role of electron spin, especially in biological life, is far more serious and central than is currently recognized. It is profoundly underestimated today. Second, the laws governing biological electricity do not always obey the same laws as the electricity flowing through wires. Third, approaches to understanding electrical processes within the organism will inevitably sooner or later be fundamentally revised. As the saying goes, take a screenshot. We'll check in a few years if we're still around. Overall, I want to say that when I look at the scientific papers published in this field over recent years, on the one hand, I am genuinely pleased to see science finally arriving at conclusions that we reached decades ago. On the other hand, however, it is regrettable that this is happening only now. But that is not the point today. However, in order to steer your thinking in the right direction regarding the nature of biological electricity, I ask you to reflect on a simple question. Do DNA and other chiral molecules conduct a spin polarized current because they possess a chiral structure? Or is their structure chiral precisely because a spin polarized current flows through them? So after this brief excursion into the nature of bioelectricity, let us now return to nanoplastics. We have established that they disrupt the electrical code of your organism at all levels. We have discussed failures at the level of ion channels and at the level of electron transport within mitochondria. Now let us speak about an even deeper injury to the electrical matrix. What specific damage inflicted by nanoplastics on cells and their components leads to the breakdown of the most subtle and vitally important process, the formation and transmission of spin polarized current? As you already understand, such a current arises in molecules that possess a chiral structure, and nanoplastics deliberately deform this structure. I will explain how this occurs using proteins as an example. This mechanism is also described in detail in many scientific studies. In biology, a protein functions only when it is correctly folded. Its shape is the foundation of its function. For many proteins, this functional form is the alpha helix, a stable structure held together by a system of hydrogen bonds. What happens when a nanoplastic particle appears nearby? The nanoplastic alters protein secondary structure. The surface of a plastic nanoparticle is electrostatically charged and chemically active. When a protein comes into contact with this surface, hydrogen bonding and electrostatic interactions immediately arise between them. And here we reach a critical moment. Nanoplastics begin to intercept the hydrogen bonds that previously held the protein in its correct form. With a protein, this presents a choice between two options, either to maintain its complex energy-intensive alpha structure or to adhere to the surface of the nanoplastic, where the overall energy of the system becomes lower. From the standpoint of physics, a protein always follows the path of energy minimization. At this point, nanoplastics effectively become a false template for the protein. The protein uses the surface of the nanoplastic as a scaffold, conforming itself to it. In other words, it reassembles and changes its shape, not because it has broken, but because the nanoplastic forces it to do so. In science, this is known as the template effect. That is, nanoplastics present the protein with an alien mold, and the protein begins to fold not in the way required for life, but in the way that is energetically advantageous in contact with the nanoplastic. As a result, the protein alters its secondary structure, and its alpha helix disintegrates. Adopting to the false template imposed by the nanoplastic, the protein is forced to reorganize into a structure known as a beta sheet. Why is this dangerous for bioelectrical processes in your body? Because a protein's function depends directly on its shape. The alpha helix has a chiral form and therefore is capable of supporting spin-dependent electron transport. When nanoplastics forcibly drive a protein from an alpha helix into a beta sheet, spin transport drops sharply, and together with it, the precision of biological processes declines. Nanoplastics also act as a catalyst. It is sufficient for them to come into contact with a single protein and alter its structure into an incorrect one, and this triggers a chain reaction of aggregation involving surrounding proteins. Distorted proteins that have altered their structure under the influence of nanoplastics lose their original program and original function and begin to aggregate with one another. This is how so-called amyloid fibrils are formed. These are precisely what are found in the brains of patients with Alzheimer's and Parkinson's disease. And it is the nanoplastics that initiate this process at the level of electrical impulses. Under normal conditions, the organism has a built-in mechanism for destroying damaged proteins, a kind of internal cleanup system, so that they do not continue to participate in cellular reactions. But here too, nanoplastics deliver another blow. They block the cleanup system itself. The cellular cleanup system consists of lysosomes, figuratively speaking, cellular waste processing plants. Their role is to destroy damaged proteins. However, nanoplastics disable lysosomes. The lysosome is literally overwhelmed by this material and ceases to perform its functions. And all of the processes I have just described occur in every cell of the body into which nanoplastics enter, including the cells of your brain. As a result, your brain shifts into a kind of energy-saving mode, mental fog, memory loss, and the fading of cognitive functions. This is not aging and not a disease. It is the result of your biological wiring, so to speak, being clogged with electrostatic noise, nanoplastics that have rewritten the shape of your proteins. We have created a material that imposes its lifeless rules on living matter, and now we are reaping the consequences of this interaction. At this stage, you understand that nanoplastics are capable of altering the secondary structure of proteins, thereby destroying their electrical and spin order. By the same principle, they also affect DNA. Studies show that nanoplastics can distort individual regions of DNA and disrupt its overall structure, literally wedging themselves into the helical spiral. Moreover, nanoplastics are capable of partially or even completely disrupting the helical organization of DNA. This inevitably leads to serious failures in electron transport and spin polarized transport, which play a key role in genome stability and repair processes. In other words, nanoplastics strike directly at the fundamental electrical mechanisms of life, not along a single pathway, but along all of them simultaneously. This is precisely why their influence has long-term consequences for health. Recently, a highly illustrative mechanism of nanoplastic interaction with DNA in the brain has been documented. I would like to dwell on this example in particular detail. Although this is a laboratory study, extrapolating its results to the processes occurring in the real human brain clearly demonstrates what happens when nanoplastics breach the electrical code of your brain. In such a state, the brain quite literally begins to lose control. In our electrical system, there is yet another level of defense that nanoplastics manage to deceive. This concerns DNA and the way the brain responds to its damage. I will illustrate this using a recent study conducted on human brain organoids, whole brain organoids. These are mini models of the developing brain that closely resemble real human biology. What did scientists observe? The nanoplastic particles used in this study, measuring 50 nanometers, penetrate the cell nucleus and trigger oxidative stress. This stress is so intense that it leads to physical breaks in the DNA helix. These are known as double-strand breaks. And this is where the most dangerous phase begins. Inside your cells, there is a sensor called TLR9 receptor, also known as toll-like receptor 9. Under normal conditions, this sensor exists to recognize the DNA of viruses and bacteria. It is a non-self detection system. But when nanoplastics tear apart your own DNA, fragments of this damaged helix end up where they do not belong. The TLR9 receptor detects these fragments and makes a fatal error. It interprets your own damaged DNA as a viral invasion. The defense system spirals out of control. A false alarm is triggered. The brain begins to attack itself. Severe neuroinflammation develops. Cytokines are released that quite literally boil brain tissue. But this is not the end. This false signal leads to a phenomenon scientists call excitotoxicity. Remember this term. It means death by overexcitation. In simple terms, your neurons begin receiving a constant demand to fire. They become so overstimulated that they quite literally burn out of electrical overload. Here, nanoplastics act like a saboteur that not only blows up the foundation, DNA, but also hacks the alarm system, TLR9, forcing the guards to fire on their own. As a consequence, the structure of neural connections disintegrates. Layers of the cerebral cortex become deformed. Cognitive abilities decline and epileptiform brain activity emerges. I hope you now understand even more clearly that nanoplastics are not merely debris in the body. They are a foreign agent, a saboteur that forces your own immune system to destroy your brain under the illusion that it is protecting it from a virus. And all of this is the result of a crude, lifeless material interfering with the most delicate electrical tuning of life within you, a material for which evolution has no defensive protocol. I have provided only a few examples illustrating how nanoplastics at different levels distort and ultimately destroy the bioelectrical order of your organism. If you wish, you can easily find dozens or even hundreds of additional confirmations. Today, an ever-growing number of scientific papers on this topic are being published, virtually on a daily basis, particularly in China. One would hope, of course, that other scientific centers, including those in the United States, would not remain on the sidelines, as this field holds the potential for major discoveries and serious consequences of a global scale. But that is not our focus today. My objective was different. It was to convey how nanoplastics hack the electrical code of your life, disrupting its very impulse. Now that you see the entire picture from the broken protein helix to the deceived immune system of the brain, the conclusion becomes obvious. Nanoplastics do not cause a single isolated malfunction. They disrupt the very logic and organization of electrical processes in your body. They intervene not where the organism can easily compensate for damage, but precisely where the fundamental rules governing the system are formed. We can now answer the central question: what exactly is being destroyed when we speak about the impact of nanoplastics on a living organism? This is not about the destruction of a single molecule, a single gene, or a single protein. It is about the violation of the rules governing coordinated system-wide operation. Life is not the sum of its components. It is a mode of operation, and it is precisely this mode that nanoplastics destroy. One can say that the electrical code of life is formed at the intersection of 3 levels. The first level is geometric. This is the chiral organization of DNA and proteins. The second level is electrical. This includes the distribution of charges, dipoles, and proton channels. The third level is quantum. This involves spin selectivity and other quantum effects that are still unknown to science. These levels mutually reinforce one another, which is why biological systems can maintain order under conditions of constant thermal noise. However, nanoplastics interfere simultaneously with all 3 of these levels. They deform geometry by physically contacting molecules and imposing false templates of form. They disrupt electrical homogeneity by creating local fields and distorting charge transport. They reduce the order and precision of bioelectrical processes that depend on chirality and spin. Why are the consequences of the intrusion of this plastic agent of chaos so difficult to detect immediately? Because the organism is capable of compensating for a decline in functional quality for a certain period of time. It increases energy expenditure, activates repair systems, and strengthens antioxidant defenses. But all of this comes at a cost, the price the organism pays to maintain outward stability. Over time, that price becomes too high. This is precisely why the effects of nanoplastic interference do not manifest as acute poisoning, but as a gradual yet active escalation of risk. Neurodegenerative processes, cognitive impairment, reproductive disorders, and genetic instability. All of these conditions share one common feature: the loss of precision in electrical signaling. Nanoplastics do not destroy only molecules or genes. They destroy the conditions under which molecules and genes operate in a coordinated manner. And this is, in essence, the destruction of the code, the electrical code of your life. In this context, it becomes eminently clear why the nanoplastics problems cannot be resolved solely through environmental measures or medical recommendations. We are dealing with a disruption of the fundamental physical conditions that underlie the functionality of living systems. This is a level at which superficial approaches are insufficient and ineffective. It is precisely here that science encounters a new challenge. We must learn not merely to measure the concentrations of nanoplastic particles, but to understand exactly how they interact with the electrical and quantum organization of living matter. We must also learn to accurately assess deeper effects, the influence of electrostatically charged nanoplastics at the quantum level, as well as the long-term consequences of this influence. I would also like to draw your esteemed attention to one more important point. We have already touched on the topic of spin and quantum nature of biological processes, areas that have been actively studied in recent decades, and that, as we believe, remain far from exhausted within the framework of the modern scientific paradigm. For many years, the illustrious scientific community has also conducted systematic research in this direction. This is precisely why we state with a high degree of confidence that science still has numerous fundamental discoveries ahead in this field. This position is based, among other things, on the fact that in our analysis, we rely on knowledge and concepts of bioelectrical and biophysical processes that have not yet received a full description or explanation within academic scientific models. In this regard, I will make a brief but fundamentally important digression to explain why we consider it possible to rely on this body of knowledge and why we are so confident in our conclusions. It is an honor for me to mention the fact that the hypothesis of the fundamental role of spin in biological electricity was proposed more than 20 years ago by my friend and colleague, Egon Cholakian. It was the same gentleman, even then, who posed a fundamental question, methodologically uncomfortable for the existing physical paradigm, yet a breakthrough in terms of understanding electrical processes in living systems. Is the electricity flowing in a living cell the same as the electricity flowing through wires? His remarkable scientific intuition guided us in a direction that very few at the time even dared to consider, toward what would later be called spin biology. Who could have known that decades later this direction would become the cutting edge of science? But at that time, for official science, it was simply too early. Here I am compelled to touch upon a topic that may be familiar to those who were engaged in science in the 1990s within the post-Soviet space. This did not directly affect me, as the core of my scientific work took place in the United States. However, I indirectly became a witness to what was happening there. What unfolded was a total paralysis of this academic system. This stood in stark contrast to the high standards set in the 1960s and 1970s when the United States had to exert a tremendous effort to avoid losing the scientific race. By contrast, in the 1990s, science in the countries of the former Union suffered a complete collapse. Bureaucracy, inertia, the stifling atmosphere, forgive the expression, of academic offices. Where in such an environment was there room for free scientific thought? Funding was allocated only to projects that promised immediate profit or were connected to oil. Everything else languished. Research gathered dust, scientists faded into obscurity, and the brilliant ideas died in desk drawers. Grants could take years to secure, and a single student's paper would be burdened with the names of 20 academics eager to attach themselves to it. It was precisely this environment that gave rise to the necessity of creating an independent scientific association, ALLATRA. This was a forced step. It was a union of enthusiasts, academics, professors, and practitioners from different fields and different countries, including the United States, who found the confines of official dogma too restrictive. People who understood that we knew only 1% of the world and that survival requires a much broader perspective. As a rule, such people are united by a shared research impulse, a deep scientific curiosity, and a sustained inner motivation towards discovery, exploration, and the expansion of the boundaries of understanding. In this context, I would like to particularly highlight the scientific vision of my colleague, Egon Cholakian, whose research intuition repeatedly proved to be ahead of the theoretical frameworks available at the time. This applies equally to questions related to the role of electron spin in bioelectrical processes, to the problem of harm caused by electrostatically charged nanoplastics, and to the geodynamic crisis. The ALLATRA scientific community began to grasp the full scale of the geodynamic danger facing our planet as early as 1995. Not a threat, 1,000 years from now, but one unfolding in the coming years. And all the assessments and projections we made have regrettably proven to be 100% accurate. Beginning in 2014, we tried to present the world with ready-made solutions, whether it was a proposal for the control of degassing of the Siberian magma plume or the introduction of atmospheric water generation technologies. We sincerely believed that we would be heard, understood, and that action would be taken. We counted on rational judgment. We naively assumed that those in power would speak so eloquently about democracy and the future were genuinely interested in the survival of their countries and their people. We were mistaken. It turned out that behind the beautiful words, there was emptiness, only business, only money. The first thing they calculated was financial loss, not human lives. The future of humanity did not concern them. As a result, we were not merely ignored, we faced persecution. That mistake cost us time, but now the situation has changed. Today's threat, nanoplastics, no longer concerns only microeconomics and, by extension, geopolitics. It concerns each of you personally. It is inside you right now. And I state this responsibly. Chemistry is powerless in this regard. You cannot chemically dissolve plastic inside a cell without killing the cell itself. This is a question of physics, specifically a question of interaction at the quantum level. Nanoplastics are nano-killers that destroy the very blueprint of life. That same spin code whose potential vulnerability we were warned about more than 20 years ago. And it is precisely because during that period, Igor Danilov sent us on the scientific path, defining the direction of our research, that today we understand where and how answers must be sought. This has allowed us to significantly deepen our understanding of the physics underlying this process. And that is why we understand that the answer to neutralizing the harm caused by nanoplastics lies somewhere here, in the domain of quantum interactions and spin orientation. But I will be frank. Currently, the Alatra group of scientists no longer possesses the resources it once had. We no longer have the ability to continue experimental or laboratory research. However, what has remained with us through years of persecution, upheaval, and loss is our voice and our knowledge. And that is something no one can take away from us. Therefore, today, with all rational people on this planet, we will not tire of defending again and again our right to life. Today, humanity must finally see the true scale of the nanoplastics problem, fully acknowledge it, and recognize that if all scientific resources are not urgently directed toward solving it, it may soon be too late, because nanoplastics, this alien presence, are already at this very moment modifying and distorting not only your electrical code, but also the electrical code of children who are still in the womb. Nanoplastics are already disabling in them the function to think, the function to reproduce, the function to live. To prevent these functions from being extinguished completely, we as humanity are obligated to find a solution as quickly as possible. The task is formulated with absolute clarity. We must find a way to deprive the enemy of power, to strip nanoplastics already present inside our bodies of their primary weapon, the ability to retain an electrostatic charge. But if we allow bureaucratic barriers and the personal financial ambitions of the powerful to rob humanity of its future, then we, the people living today, risk entering history as the last generation of humans on this planet that was capable of clear thought and relative health. But I believe that we as humanity have sufficient motivation to live, because life is truly worth fighting for.